Now we will work on topic 8, transport in plants. We have learned everything about plant nutrition, photosynthesis, limiting factors, the route or the fate of oxygen, and the leaf structure and function of every single layer. Now we need to talk about the transport structure of flowering plants. This is the objective of the first objective um, for the first part of the lesson. Uh, we need to state the functions of the two types of transport tissues in plants. Xylem and phloem are the two types of tissues responsible for transporting all of the materials needed for plant. Definitely, there is a difference between xylem tissue and phloem tissue in structure and in function. Let's go through the PowerPoint presentation in order to understand. The functions of xylem and phloem is to transport all of the materials inside the plant. So xylem to conduct or transports water and mineral ions. And very important to know that plant absorbs water and minerals by root hair cells, but xylem transports. So very important, guys, to use the accurate verb in the accurate place. Xylem transports or conducts, but root hair cells responsible for absorbing all of the materials from the soil. We will talk about the absorbing or the absorption process because water will be absorbed by osmosis, but mineral ions by active transport. Let's go through this while answering questions, but now we need to talk about the function of the phloem tissue. Phloem tissue conducts all of the food made in the leaf to all parts of the plant. So take a look on the xylem vessel and on the phloem tissue. You will find a lot of differences. Xylem tissue considered as a dead tissue because it has no cytoplasm, it has no cross walls, it has no cross ends, it has a thick layer of lignin to support the tissue or the stem tissue. So a lot of modifications happen in the xylem tissue in order to transport water from the root to the stem, leaves, and the flower. On the other side, phloem is a living tissue. It has cytoplasm. It has companion cell. Companion cell, it means cell attached to these phloem vessels or the sieve tubes, and it has all of the materials needed for the cell. So phloem considered as um, living tissue or living structure, but xylem considered as a dead tissue. How xylem vessels adapted for its function? Look deeply on xylem vessel. You will find that it has a thick layer of cellulose, has a secondary thick wall, has a spiral bands of lignin, has pits, and those pits responsible for communication channels in between xylem tissues, and it is hollow in order to absorb all of the water up. One force of the forces take water up is the adhesion and cohesion force of water molecule. The attraction between water molecules itself help to rise water up, and the attraction between water and the walls of the xylem vessel, and this is called adhesion force, helps to take water up as well. So cohesion and adhesion forces are extremely important to transport water. This is the structure of the phloem. So I mentioned that we have sieve tubes and sieve plates, and all of the sieve plates um, has sieve plates have a lot of minute pores to allow rapid flow of food substances. So very important to know. And if we took the TS or transverse section, you will find the sieve plate and you will find the companion cell. This is a lateral cell or attached cell to the sieve plate cell. How are the vascular tissues organized in stem? Oh, this is an important point. Xylem and phloem arranged similarly in the stem and root tissues in dicotyledon plants. In the stem, xylem arranged in vascular bundles like this, arranged in a circle, okay? And xylem looks inward, okay? So xylem looks inward, but phloem look outward or located outward. This is in the stem and also in the root. So you will ask me definitely a question, sir, how could we identify xylem vessels or xylem tissue and phloem tissue? Number one, the location, as I told you, unchangeable, and the shape and the organization of the tissues, like those types of tissue. Okay, so this is the xylem tissue and this is the phloem tissue. So xylem and phloem tissue arranged in that way. Okay, so el phloem upward is xylem inward in stem and roots. In the root, the shape is a bit different, but still we are highly stick to the rule. Xylem inward and phloem outward. It's like a cross shape, but this is the phloem and this is the xylem tissue. In the leaf structure, the arrangement is different. Xylem is located upward and phloem inward. So please, Alex, don't forget that the arrangement of xylem and phloem will make a difference in your answer. The movement of substances in plants. Very important part, and the majority of this part is a supplement or extended part. How can we study the path water takes through a plant? We can carry out an experiment by preparing or designing this setup to study the path of water through a plant. We will add red ink diluted water like this, and we will check after a while how this diluted red ink transported in the plant. It will be transported through xylem, through phloem, through leaf, through stem, through root. We will check this after a while. So this is an amazing experiment, and we did it in the school lab, and you cannot imagine the color and the absorption and the transport of water from the root hair or even from the stem if there is no root by capillary across the xylem vessel. How can we study the path food substances take through a plant? We will check this by using any kind of aphid feeding on the carbohydrate or the carbohydrate stuff, and we will check that the carbohydrate stuff, when the needle-like structure or this feeding style reach the phloem, so we will definitely detect the phloem tissue and how it could transport all of the sucrose or starch sucrose only to be stored in a form of starch in the plant. So by this method, we can definitely study something called translocation. Translocation. I will talk about translocation. Um, let's talk about translocation now. Look, Alex, in the plant, we have leaf like this one, and we have some roots like those roots. Okay, so if the leaf or this part of the plant can carry out photosynthesis process, we named this part as the source because now it is the source of food. If this part, and now this part called the sink. So if the leaf will carry out the photosynthesis process, it considered as the source, named as the source, and the roots, which stored all of the materials, named as a sink. For any kind of plant under any unsuitable conditions cannot carry out photosynthesis process, so we could absorb some of the materials or take some of the materials stored before from the roots, which now will be the source because it became the source of food, and we can transport all of these material throughout all parts of the plant, including leaves, and the leaf now will be the sink. So same structure could be sometimes source and sometimes sink. This is called translocation or exchanging the role in the plant. You got it? So translocation is when plant parts exchange the role by being source if they can carry out photosynthesis, or if there is no need to mention photosynthesis, if they are the main parts of food. This question is also important because we found this question recently in a lot of exams. If we cut some tissues in specific part of the plant, if we cut phloem, definitely plants cannot transport all of the materials throughout the plant or the phloem tissue. So if the source is the leaf, so there will be swelling here in this area, but materials will not be transported. Okay, so very important to know. We cut the plant in what area and what type of tissue has been removed in order to answer this question. This is the slide of the source and sink. You can read it and check everything after the session. And now we need to talk about movement of water. For movement of water, a lot of objectives, but all of them are extremely easy. How does water enter a plant? Now we need to talk about root hair cell and water movement or absorption of water occurs by osmosis. Osmosis is the net movement of water from high water potential to low water potential. So water will be absorbed now through root hair cells, going directly from high water potential to low water potential in the cortex layer until it reaches the xylem vessel to be transported easily. In the exam, can we take the 4 marks in our answers? Definitely. Definitely, because you will talk about osmosis, you will talk about absorption of water from high water potential to low water potential through partially permeable membrane. This process does not require energy, and you will mention after that the layers that the water passes through. How does water enter the plant? I think we answered this question and we mentioned everything. For transpiration, transpiration is the loss of water vapor from the aerial parts of the plant. So through stomata of the leaves and the lower epidermis, water will be lost. Water will be lost in a form of water vapor. So you cannot say that water will be lost by osmosis. This will be a huge mistake and you will lose the mark. Water vapor will be moved by diffusion because water vapor is a gas. And I mentioned this several times during the explanation. Look at this experiment. More water droplets are seen on the inner surface of the bell jar with the leafy twig because leaves will transpire and will release more water vapor from the lower epidermis. On the other side, there is no water droplets on this bell jar because there is no leaves. Look at this drawing, and I like the idea, or I like the fact that the drawing will give you all of the points needed to take the full mark or to achieve the best results. Why? Because he might ask you a question like this: how is transpiration involved in moving water against gravity? So we can say that water will move by osmosis from high water potential to low water potential when there is a concentration gradient in the leaf. So based on the concentration gradient in the leaf, we can definitely absorb more water from the soil. Did you understand this, Alex? If we are making a concentration gradient because there is a loss of water from the leaf, so there will be less water potential and high water potential, so water will move to the next cell, to the next cell, to the next cell. So water will be absorbed already from the soil. So without transpiration, we cannot absorb more water from the soil. And here we are talking about movement of water inside the leaf, and I think it became now something primitive because water will move inside the leaf through the air space, through the guard cells, and from the xylem to the air space and to the guard cell and stomata. I think it became extremely easy now. Look at this. I think the importance of transpiration is totally clear. Transpiration draws water and mineral ions from the root. Without the concentration gradient or the concentration difference, we cannot absorb more water at all, because we are absorbing more water if there is a concentration gradient. Evaporation, we cannot cool the plant if we cannot release or lose more water. Water also transported to the leaves can be used in photosynthesis. So in order to keep cells turgid and to replace water lost by the plant, we have to transport more water to the leaves. This is the importance of transpiration. This potometer or potometer is used to investigate the rate of transpiration. Usually, we will put shoot part because shoot part will transpire, and based on the distance moved on this graduated capillary tube, we can measure the rate of photosynthesis definitely. We have several factors affecting rate of photosynthesis. So, rate of transpiration, such as temperature, definitely. And there is a positive correlation as temperature increase, rate of transpiration or losing water from the plant increases. The second factor is the wind speed. As wind speed increases, rate of transpiration increases as well. The third factor is humidity. Humidity, it means the amount of water around the plant, around the shoot part. If we have more humid atmospheric air, this means that the transpiration will be lost. And the opposite, definitely. If we have if we have less humidity, this means that we have more transpiration. Okay, so we can consider those as the factors of uh, transpiration. Don't forget that when we have light, we have photosynthesis, so we have more water to be absorbed by the plant and more heat energy released, so we will lose more water as in a form of water vapor. So light definitely increase the rate of transpiration. This is the transpiration topic, guys. Please check the concept map and or design your concept map if you feel like you cannot understand this map. Please, please, please draw this and work on this in order to get the best results and any question related to this topic, the plant transport topic.